Okay, well, welcome everybody to our webinar series for HIRN. So glad you could join us today. I'm Joyce Nyland. I'm a co-PI of the HIREC, the Human Islet Research Enhancement Center of HIRN, and also a professor and department chair at City of Hope in the Department of Diabetes and Cancer Discovery Science. Our lecture today, our webinar, will be an exciting reveal, a new introduction to the new Islet function portal on PANC-DB, one of the major database resources of HIRN. It's going to be presented by Jeffrey Roman and Nilanjana Samanta from the University of Pennsylvania, where the PANC-DB has been developed under one of the consortia of HIRN, the HPAC consortium. Just want to give you a little introduction for those of you new to HIRN, and a little bit about today's webinar. So HIRN is an evolving ecosystem of scientists across the nation who started out as these several consortia that you see here, along with a coordinating center and a bioinformatics center. And then as science changes and the funding sources evolve, it's changed now into these 5 various consortia. We still have beta cell death and survival, and HPAC, which is where this system PANC-DB arises, the Human Pancreas Analysis Consortium. We also have now CMAD, the Consortium of Modeling Autoimmune Diabetes, and PANC-BASE is one of the newest consortia you'll probably hear about in a future webinar, where they're trying to integrate data across all of these various sources. We're waiting for a logo for CMAD. That's why that one is purposely blank so far. This consortia has grown over the years. We're now in our 11th year. We're tracking the publications that are generated by our researchers in HIRN. There's been over 1,000 so far. For the 800 researchers that have participated, at this time there's about 200 or more current researchers, and that spans 109 institutions across 12 different countries. So it's a continually growing and evolving set of research consortia. One of the things I'd like you to just be aware of is the HIRN newsletter. You can sign up for it and get it automatically in your inbox, and it'll help you know about HIRN members and manuscripts, upcoming meetings and webinars such as these. Job opportunities are posted there, and importantly, new funding initiatives will appear in the HIRN newsletter, so you can learn about how you might apply for funds to support your diabetes research. For example, right now, there are a couple coming up. One's called the HIRN Data Scholar Awards. This is a new funding initiative that will support using existing HIRN data sets and resources to explore unique, innovative hypotheses and accelerate breakthroughs and advances in T1D. These are data science-centered projects that will be submitted through us as the HIREC. They'll use existing data sets from HIRN and related T1D programs to create and design studies and resources. And it'll be an opportunity to collaborate with HIRN investigators, exchange ideas and resources, and help us accelerate T1D research. The next application due date is November 1st, 2025. And then there's the Investigator Gateway Awards for collaborative T1D research. These support a robust pipeline of innovative projects, talented new investigators. The awardees are embedded within established scientific frameworks in an existing consortium. It may be this one or TrialNet or TEDdy. It'll provide unique opportunities for new and early stage investigators. So it'll propel their career and increase their understanding of key questions and benefit HIRN by providing these new ideas and perspectives. The next due date for this application is March 6th of 2026. And then you should be aware of our website. The URL is given up in the corner there, hirnnetwork.org. And here you'll find a number of various resources as well. The funding opportunities are announced here, and other newsletters can be accessed. Resources such as the HPAC PANC-DB we're going to hear about today, the IADP, which I also run that for islet distribution, NPOD, etc. And we also have our social media. You can follow us on social media. The resources that we curate for diabetes research have now grown to over 3,400 different resources, antibodies, cells, data sets. So take a look. These are all free and publicly available out on our website. And also, just be aware, all the webinars that we've given and this one today are posted on our website on the YouTube channel, so you can go back or share it with your colleagues. So for today's talk, just a few little reminders. This presentation is being recorded and will be posted on YouTube. Please mute your audio and turn off your video during the presentation. But at the end, discussion and QA will follow. Feel free to turn on your camera, raise your hand, or use the chat box. And the topics that are going to be discussed today are an overview of islet perifusion and related physiology datasets that are available on PancDB. And then Nilanjana is going to give a live demonstration of the new interactive data analysis portal for islet perifusion. So I believe that Jeffrey will begin and give you a little bit about the perifusion and physiology. Jeffrey, I will stop sharing, and if you guys want to share your screen now.
Thank you so much, Joyce. So I'm going to share my screen now. Okay. Let me know if you're able to see
Yes.
my window, the slide.
Yes, that's good. If you want to put it in slide mode.
Yes.
Great, there it is. Thank you.
Great. All right. Thank you so much for the introduction, Joyce. So good afternoon, everyone. Welcome to today's webinar. Thank you, HIRN, for the opportunity. I am Nilanjana Samanta. I am the data curator for the pancreatic database, or PANC-DB. And my co-presenter today is Jeffrey Roman. He has over 10 years of experience building perifusion systems for different disease models and joined the HPAP team about 5 years ago. Together, both of us today are going to introduce to you a newly launched interactive analysis portal for islet function data that's already available live and ready for you all to explore. So this is a rough outline of our talk today. I'll give you a very small overview of HPAP and PancDB. Jeff will then take over and familiarize you with the islet physiology data that's currently there on PancDB. And then I will finally come over and then give you a live demonstration of not just the portal that we're talking about, but also how to access the raw data and download it. So to begin with, if I have to describe HPAP in one sentence, it would be that it is a multi-institutional effort comprising of experts from all of these different institutions. What you see here is the operational workflow of HPAP, and PancDB is the public-facing entity. We have an array of imaging, genomic, epigenomic, islet function, and immune profiling data. All of this data are publicly accessible on PancDB. All of this data is generated from not just pancreatic tissues and islets, but one thing that makes us very unique is the availability of immune profiling data generated from peripancreatic lymph nodes and spleen, in addition to the more traditional approach of using peripheral blood. So all of this makes PancDB the largest database for original multimodal data for both T1D and T2D, that is type 1 diabetes and type 2 diabetes donors, as well as autoantibody positive donors. You might have seen our HPAP data in some other databases or knowledge bases, like Joyce mentioned, the PancBase. Know that this is where the original data is coming from. But today's focus is the islet physiology data. It is contributed by 3 institutions, one being Penn, the team that Jeff is a part of. The others are Vanderbilt and Alberta. But for today's webinar, we are going to focus on the protocol that the team at Penn uses. To give you an overview of the data we currently host, we have about 25 terabytes of data from 186 donors at present. All of these donors can be grouped into these three major categories, that is type 1 and type 2 diabetes and non-diabetic donors. These non-diabetic donors can be further subdivided into a group called the autoantibody positive donors. These donors are basically positive for a different combination of these four autoantibodies that are crucial for detection of type 1 diabetes, as you can see in this bubble plot here. So now Jeff is going to take over and talk to you about the Islet perifusion data and some other data sets on PancDB.
Thank you very much, Nilanjana. Hi, everyone. I know that we have a lot of islet biologists in the audience today, but we also have some people who work strictly with data, as well as members of the general public who may be watching live today or in the future on YouTube. So I'll give a very brief introduction into human islet biology, as well as the perifusion technique which we use for HPAP to assess insulin and glucagon secretion from human pancreatic islets. So very generally, we know that the pancreas is the organ which helps to maintain blood glucose levels at normal. And the way that it does that is through these little clusters of cells. These are called the islets of Langerhans, or islets for short. You can see an image of a single human islet here on the top left, which is stained for the different cell types in the islet. Now, the human islet is comprised of multiple different cell types, the chief of which is the pancreatic beta cell. Now, the role of the pancreatic beta cell is to secrete insulin when blood glucose is high. Insulin is the signal that tells the rest of the body that glucose is abundant and to uptake glucose. Another very important cell type, which is gaining attention very recently especially, is the pancreatic alpha cell. And the role of the alpha cell is to secrete glucagon when blood glucose is low. So you can imagine, let's say you eat a meal, your blood sugar increases. The beta cells can then sense that your blood sugar is elevated, will secrete insulin into the bloodstream, and through the bloodstream, it's delivered to the rest of the body, tells the other cells in the body to consume glucose. And as those other cells consume glucose, your blood glucose will drop naturally to normal levels. On the other hand, let's say you haven't had a meal in a while and your blood glucose is low. Now the alpha cells will sense that your blood glucose is low and they will secrete glucagon. This will tell the liver specifically to stimulate glucose production. And specifically, this is by breaking down glycogen. And in this way, both the beta cells and the alpha cells fine-tune their insulin and glucagon release to help maintain blood glucose homeostasis. There are other cell types in human islets, for example, delta cells and pancreatic polypeptide cells. But by and large, as you can see in this panel top right, that the insulin-secreting beta cells and the glucagon-secreting alpha cells comprise the vast majority of human islets. And so when we talk about human islet function, what we're really talking about is insulin release. How much insulin do they release? How much glucagon do they release? So this is what we measure through our technique, which is called perifusion. Now, perifusion, relatively simple. What we do is we take human islets, these are isolated human islets, and place them inside a chamber. This is called the perifusion chamber. And this chamber is connected to a flow system with two very important lines, the first of which is the inflow line on the bottom here and the outflow line at the top here. Now, from the inflow line, we supply nutrients, things like amino acids, glucose, oxygen. And so we send those in through the inflow line. The human islets can see this glucose and amino acids, and then they secrete insulin and glucagon in response. And through the outflow line, we collect that insulin and glucagon. And then by radioimmunoassay, we can measure how much insulin and glucagon the islets have secreted in response to the different secretagogues. We can draw these very nice plots of insulin and glucagon secretion. These are temporal dynamics, so we can tell at any time point, based on the stimulus that we have provided, how much insulin or glucagon the human islets have secreted. So this is a picture of the actual system, the actual perifusion system, which is used for HPAP. Now, it's comprised of three major components, the first of which is a low-pressure chromatography system. Now, the system wasn't originally built for perifusion, but we have repurposed it for perifusion purposes, specifically for two key components. one of which is the peristaltic pump, which helps to maintain a very steady flow rate. And the second is a 5-way intake valve. And this allows us to introduce different substrates into the buffer. For example, we want one bottle with glucose in it, one bottle with amino acids, one bottle with a perifusion buffer with no substrate. And we can program this to deliver glucose, to deliver amino acids at very specific time points. We have in the middle here a water bath. This maintains everything at 37 degrees Celsius for optimal function of the islets. As well, we have a perfusion chamber. You can't see it here, but it's tucked inside the water bath. And this contains 1,000 human islets, which are placed on a nylon mesh filter. On the right side, we have a fraction collector. This is what actually collects the outflow insulin and glucagon. We collect these in glass tubes at 1-minute intervals. So we have minute-by-minute resolution of insulin and glucagon release through the duration of the perfusion run. And so we have a perifusion protocol which we can use to assess islet function. And this protocol is very special because it can assess the insulin and glucagon secretion. And so we can assess their function through this protocol. First step, as you can see here on the left side, is the baseline. This is where we just established basal secretion. This is without any glucose, without any amino acids. How much insulin and glucagon are these human islets secreting? Our next step is to add a physiological amino acid mixture, or AAM. And this has the primary function of stimulating glucagon secretion from the alpha cell. So you can see here, once we add amino acids, you get very nice stimulation of glucagon secretion. And so, in fact, amino acids are the primary stimulus of alpha cells. Our third step is to add glucose. This is labeled here, G3 for 3 millimolar glucose, G16.7 for 16.7 millimolar glucose. This has opposite effects on alpha and beta cells. On alpha cells, what you'll see here in red is that glucagon secretion is suppressed by glucose, both low and high. But in the beta cells, you can see from insulin secretion in the black, glucose very strongly stimulates insulin secretion. So you can see here under G16.7, which again means 16.7 millimolar glucose, you see very nice stimulation of insulin secretion. Our next step is to add IBMX. This is a phosphodiesterase inhibitor. It maximally raises cyclic AMP levels and will potentiate both insulin and glucagon release. So you can see once we add the IBMX, both insulin and glucagon release are potentiated. Then we wash out all the substrate. You can see here G0. This means 0 millimolar glucose. Reestablishes the baseline before we introduce potassium chloride. Now this will depolarize the cells and allow the islets to secrete all of the readily releasable pool of secretory granules. So you see very sharp secretion of both insulin and glucagon once we introduce IBMX. So again, this protocol is used to assess the function of both alpha and beta cells. Now, let's say we want to quantify these effects. Let's say we're looking here at an insulin trace, and we want to see what is the effect of G16.7. So it's similar to taking a sugar tablet. Let's say you take the sugar tablet. What is the effect of the sugar tablet on your insulin secretion? And we have two main ways to quantify this, the first of which is area under curve, or AUC. The way that we do this is, first I've blown up here the G16.7 portion of this plot. You can see there's some level of pre-stimulus secretion, labeled in red. And then after we introduce the high glucose, we have a post-stimulus secretion. And the area under curve is quite literally the area under the curve generated by post-stimulus secretion. So you can see the formula here, area under curve is equal to post-stimulus secretion minus pre-stimulus secretion. It is very important that we include this term here, minus pre-stimulus secretion, because we want to isolate the effect of the high glucose. We want to isolate the effect of the sugar tablet. And so we include this here so that we can remove the contribution of things like amino acids, which were added earlier. And important note here, if post-stimulus secretion is greater than pre-stimulus secretion, then of course the area under curve will be a positive number, greater than 0, which means that secretion was stimulated. On the other hand, you could imagine after we add our intervention, post-stimulus secretion ends up lower than it was pre-stimulus. In that case, the area under curve will be negative, and we say that secretion is suppressed. The other measure that we have to quantify the secretory response is the stimulation index, or SI. And so here I have the same trace. The way that we calculate SI is by taking the maximal point post-stimulus secretion and dividing it by our same baseline pre-stimulus. So we have here the formula SI equals post-stimulus secretion over pre-stimulus secretion. This quantifies essentially the fold change in secretion, which is due to the intervention. Again, I will note, if post-stimulus secretion is greater than pre-stimulus secretion, then your SI will be greater than 1. It means secretion was stimulated. And then if, on the other hand, your post-stimulus secretion is actually less than it was pre-stimulus, then SI will be less than 1, say secretion is suppressed. So now that I've gone over the facets of perifusion data, here are the actual perifusion data sets. This is what they look like if you download them on PancDB. These are CSV files organized by time point, where each row is a particular time point, and we have different columns here. You can see the time in minutes. Next to that is the stimulus that was present inside the buffer solution. Then you have the measured insulin and glucagon release in the units nanograms per 100 islets per minute or picograms per 100 islets per minute, respectively. But we also have 3 measures here, which we collect for each set of islets that we run. These are the DNA, insulin, and glucagon content. And so we extract these from the islets after perifusion. We provide them so that you can normalize your data. For example, you see here we've labeled the insulin release nanograms per 100 islets per minute. Let's say you don't want to normalize per 100 islets. You want to normalize per total insulin content. Because, for example, let's say type 1 diabetic islets, they don't have any beta cells left. There are very few remaining. They don't have very much insulin. So their insulin release will be very low. You could normalize that to insulin content to get the percentage of insulin that the islets had released. You can do the same for glucagon. Now, speaking of these normalizations, we have 2 additional normalizations that we're hoping to add by the end of the year. These are per IEQ, or islet equivalent, and also per alpha and beta cell. So for per IEQ, what this is essentially a measure of the volume, the islet volume, or the islet mass that is present in our islets, which we use for perifusion. And the way that works, we can take an image, and there's a software which can detect the things which are islet, the things which are not islet. And then for the things which are islet, calculate essentially the total volume. And so we have measures of IEQ. This is a way that we can normalize because islets don't all come in the same size. Some are smaller, some are larger. We also have normalization, which was recently developed by our collaborators over in New Valdor's lab, based on a DNA methylation method where you can detect patterns to essentially quantify the number of alpha cells and beta cells in our islet preparations. So you can normalize insulin secretion per beta cell now, or glucagon secretion per alpha cell. And so these are also very important normalization parameters. And so we will have, again, in the very near future, expanded data sets with additional normalizations. So instead of just the insulin release and glucagon release in the units 100 islets per minute, now we'll have insulin and glucagon secretion per total content, per IEQ, per alpha cell, per beta cell. And we will also supply the numbers which we used for both IEQ and alpha and beta cell percentages to calculate these numbers. So very briefly, I will go over 2 other functional tests that the perifusion core does, because not only is perifusion very important, not only is insulin and glucagon secretion important in these islets, but there are also some intermediate markers that we can use to assess islet function. The first of which is calcium imaging. We do this by fluorescence microscopy using Fura 2 dye. And so you can see here the areas which are very bright colored. These are areas of high intracellular calcium. And so we can do these traces of intracellular calcium over time. I'll draw your attention here to the non-diabetic donor on the bottom left. You can see that when we add 3 millimolar glucose and 16.7 millimolar glucose, the non-diabetic donor islets respond by increasing calcium uptake. Whereas in type 1 diabetic donor islets, as I mentioned before, type 1 diabetic donors don't have very many beta cells left. You can see that 3 millimolar glucose and 16.7 millimolar glucose elicit no response. It's completely flat. And that's because, again, type 1 diabetic donor islets do not have beta cells. We have another test here. This is oxygen consumption by quenching phosphorescence. All cells in the body consume oxygen, and islets are no exception. And so the way that we measure this is with a perifusion system, very simple to what I had shown previously. But in the outflow, we couple this to a fiber optic cable system. And what this allows us to do is very precisely measure the amount of oxygen that is present in the outflow. So you could imagine if we send 21% oxygen to the islets, they consume some amount, and we measure, let's say, 19% oxygen. Then that 2% is what was consumed by the islets. And so we can draw these very nice traces of oxygen consumption over time. Again, let me draw your attention to the control non-diabetic donors. you can see that in response to G3 and G16.7, you get very nice stimulation of oxygen consumption. Whereas in the type 1 diabetic donors in red, you see when we add both low and high glucose, the type 1 diabetic islets do not respond. And the reason is because they don't have any beta cells. Now I've included one more group here. This is very important. These are GAD-positive donors. They are positive for an antibody against GAD. This is a biomarker for type 1 diabetes progression. If you have the GAD autoantibody, you are far more likely to develop type 1 diabetes relative to the general population, even though the beta cells are totally healthy at this point still. But what you can see through oxygen consumption is that already in the GAD-positive pre-diabetic stage, that there's some oxygen consumption phenotype intermediate between controls and type 1s, indicating that there's some progression towards disease in the GAD-positive donors. This will be very important, the GAD-positive stage, and we'll talk about this a little bit more later on in the presentation. But for now, this concludes my portion, and I'll hand it back over to Nilanjana, who will do a live demo now of our new perifusion analysis portal, so that you can take a look at the data and answer some very interesting and clinically relevant questions. Thank you.
Thank you so much, Jeff, for such a thorough explanation of the islet functional data. So now I am going to go over PancDB, show you how you can access the raw data and download them. And of course, also show you step-by-step the utility of the new Islet Perifusion data analysis portal. But if you want to come join along with me, you can Google the letters PancDB or just take this URL on your browser, hpap.pmax.upenn.edu, and you will land on PancDB. And this will allow you to explore the website along with me. So now I'm going to switch over to the website. And in the interest of time, I have all the tabs open here already. Just in case anything is not properly visible to you, if you need me to zoom in or out, please feel free to let me know. So let's begin with the homepage. So once you search for our database, this is the first page you will land on. And right off the bat, you can see there are these 4 major tabs. These are the primary data exploration options that PancDB provides. If you scroll down a little bit, you will see this very simple plot, which basically shows you how much data we have for all of these different assays on the y-axis. And you can clearly see Islet Perifusion is one of the most abundantly available data set on PancDB. Now, quickly, so all of these 4 major options are also available on the top navigation bar over here. This navigation bar will be with you as you continue to explore the website and open more and more pages. Now, if you're interested in watching video tutorials on some of our on how to use PancDB, how to download the data, and some of the various applications we have, you can watch them over here on this page. If you're interested in downloading the protocol that Jeff just talked about, you can come to the workflow and protocol page, navigate to the appropriate section. For example, here, I'm here at the Islet Physiology Studies, and then you can download it as a PDF. Now let's look at how you can download the raw data. In this page, I'm going to use a very simple use case. Perhaps you're interested in downloading the Islet Perifusion data for type 1 diabetes donor and non-diabetic donor. And keeping in mind, since we are focusing on everything that the team at PEN, the protocol that PEN is using, I have selected the appropriate filter on the left side over here. So the counts you're seeing here is a reflection of my selections on the left. So now let's come over to the matrix. Here you can see all of the assays being highlighted as I hover over are listed on the top x-axis over here. They are all grouped by their cell and tissue type of origin on in the bottom over here on the y-axis. I'm interested in downloading all of the Islet Perifusion data generated by University of Pennsylvania for type 1 diabetic and non-diabetic donors. So I click that assay, As you can see, everything that's available for that assay for all donors gets selected and gets highlighted. And here is the count. 102 donors have this data right now on PancDB. And then from here, these two buttons will get highlighted. So here you can either do a batch download of everything using a bash script. And since you can, on top right over here, you can see the total size of the data volume that you will be downloading. It's less than a GB, so you can also just do a direct download. This download package will also comprise of metadata files to help you contextualize this assay. Now, if you are interested in a more granular breakdown of data availability by donor, then you would come to this by data status and donor page. Again, notice that the filters I selected in the previous page are still maintained over here. Same type 1 diabetes and non-diabetic donor, University of Pennsylvania, and I am interested in downloading islet perifusion data. So I go ahead and select that assay. As you can see, as soon as I do that, every donor that has this assay gets highlighted in yellow. And any donor that does not have this assay does not get highlighted. And for such donors, we also provide explanation on why the data will not be available. So this page is useful because, as you can see, the nature of HPAP, the nature of PANC-DB is that it's a dynamic database. So all of these panels of experiments are being conducted by the different institutions simultaneously. So they all have a different timeline. They are being uploaded to PancDB at different rates. So there is usually a delay in the data availability. And there are at times in between all of this process, there can be some technical issues like lack of samples being collected, biosamples being collected from a donor, or experiment failure. So these are all situations beyond our control, and which is why, to show you exactly at what stage, to communicate to you at what stage of data availability a particular data set is for each donor, we have this page, and we use these 4 self-explanatory data labels to communicate that to you. So now, and there is one final way you can download data from PancDB. For example, you are interested in downloading everything, not just islet perifusion data, but everything that we have for HPAP 012, this one donor. As you can see, this donor has a total of 288 gigabytes of data. You can come to the by donor list and then select everything. Or if you're interested in a single file, you can then click on that single file, and then all of your selections will get downloaded using the button over here. And then you can also download the metadata for all of the assays that you've selected. So quickly note that all of these 3 data download options that I showed you are present on the same page. That is the experiment data download page, and you're not going to different windows. They're all different tabs on the same page. Now, I've showed you that PancDB is a primary source of multimodal data. It allows you to perform data download. But in addition to being a data download portal, we are also a data analysis portal because we have developed or we have implemented various tools for our existing data. To access all of those tools, you would come to the interactive analysis page over here. And then the tools that I'm talking about, the various applications for different data types like bulk sequencing data or single cell RNA-seq data, they're all available on this page. And one such tool is the interactive islet function data analysis tool, which is the topic of discussion today. So you would access that using this button over here. Once you open that application, this is where you will land. Right off the bat, you can see this insulin and glucagon release plot, which is a dynamic version of the static plot that Jeff already showed you. He also showed you that this experiment measures minute by minute values of insulin and glucagon secretion, which you can see clearly as you hover over the plot. Now let's go over some basic functionalities of this portal. To begin, with the most basic one. If you're interested in only seeing the insulin secretion values, you would go ahead and unclick the glucagon option on top over here. Similarly, if you're only interested in viewing the glucagon release, you would go ahead and unselect the insulin option. If you're interested in downloading this plot as an image, you can do that using this PNG button over here. If you're interested in downloading the raw data like Jeff showed you previously, the CSV file, you can do that over here. This will be for the donor that you're looking at over here. And right on top of the plot, we provide some basic clinical information to contextualize what you're viewing. On the left over here are a set of important demographic and clinical information that will allow you to correlate these to the islet perifusion data. And so basically here I have selected for the disease type 1 diabetes, because I'm interested in looking at the traces, insulin and or glucagon traces just for type 1 diabetic donor. As soon as I do that, as you can see, I have a total of 72 donors available to view on this portal, but I have 10 type 1 diabetic plots. So now these 10 donors will get listed in this drop-down list over here. But I'm interested in 1 or 2 donors that I want to come back to and do a comparison. So for example, here I'm interested in HPAP 135. So I have tagged that donor. And I'm also interested in comparing it with some other donors. For example, HPAP 002. So I will go ahead and tag that donor as well. So as you can see, both of my tagged donors are now listed below over here. So I can now go back to HPAP 135, look at its insulin trace, and then compare it to HPAP 002 very easily and look at that donor's insulin trace as well. Now, as you scroll down, you will see these 2 buttons. So these are nothing but the option to download how many ever CSV files, the raw data for islet perifusion. So here you can see, based on our selection, you can download the data for all 10 of the type 1 diabetic donors together. Or if you're only interested in downloading the data for just these 2 tagged donors, you can do that using this button over here, download data for tagged donors. You will remember from one of Jeff's slides where he showed you how to calculate the summary statistics, that is the area under the curve and the stimulation index that quantify islet function for all of the 5 stimuli that are introduced during the experiment. Those absolute values are calculated for every donor for both insulin and glucagon, and those are the values I'm showing you are presented in this table over here. And then at the bottom, these values, the area under the curve and stimulation index, the absolute values for each donor are plotted against the rest of the HPAP cohort for each one of these 5 stimuli that you can see over here. As you can see, this HPAP, one of the HPAP 002, that is one of my donor of interest, who is also type 1 diabetic, as expected, falls in an extremely lower percentile compared to the rest of the HPAP cohort in terms of insulin secretion and in terms of response to high glucose stimulation. Now let's go over this portal. Let's look at its utility using a more practical example. For that, I'm going to build on something Jeff already talked about in his last slide. He mentioned that individuals that are GADA positive are more likely to end up developing type 1 diabetes in their lifetime compared to the general population. However, not all of them end up developing type 1 diabetes. So keeping that in mind, I have gone ahead and selected these filters where the donors are GADA positive but not positive for any other autoantibodies. I also want to select non-diabetic donors because I'm interested in looking at the likelihood of them developing type 1 diabetes later. So I've gone ahead and selected non-diabetic donors. And then I also want these donors to be age matched. So I've selected a specific age range between 20 to 30 years old. They are not pediatric. They're also not geriatric. They are somewhere in between a healthy set of donors. So now I've gone ahead and selected 2 random donors in this who fall under these parameters that I've selected. So the first donor is HPAP49. I'm going to go ahead and unselect glucagon because I'm only interested in their insulin secretion. If you look at the blue plot, the y-axis, you can see that this donor has somewhat of a good amount of insulin secretion. It goes all the way up to 10 nanogram per 100 islets. If you scroll down, let's look at where it stands compared to the rest of the HPAP cohort. It's pretty good. It's higher than 85% of the HPAP cohort. Now let's look at the next donor, HPAP50. So HPAP50, if you look at the y-axis here, has extremely low insulin release. It's less than 1 nanogram per 100 islets. Compared to HPAP49, that for whom the insulin secretion went all the way up to 10. Now, if you look at this cumulative distribution plot, you can see that this donor HPAP50 is producing insulin only more than 7% of the donor, so it's extremely low. So this, to me, it looks like, and I think it's safe to say that this HPAP50 is behaving more like a type 1 diabetes donor, whereas HPAP49 is behaving more like a healthy donor. So clearly, these 2 donors are clinically very similar, but there is something that islet perifusion experiment is picking up on, some physiological difference in islet function. Now, most of you know that islet, the alpha and beta cells, let's talk about the beta cells. Type 1 diabetes donors have very few beta cells left at the first place. So could difference in cell count answer this physiological difference that we are observing? So for that, I have come back to this donor information page on PancDB. Here you can get a snapshot of important clinical information. And one of them is the endocrine cell composition for all these donors. You can see clearly that HPAP49 that was behaving more normally and HPAP50 that was behaving more like a type 1 diabetes have an extremely similar alpha and beta cell distribution. So this alone cannot answer, is unable to answer the physiological difference in islet data that we are observing in these 2 donors. Perhaps these donors have a genetic predisposition. Maybe we can answer this genotypically. So to analyze that, we have another interactive analysis portal, which essentially shows you the genetic risk, the overall genetic risk for developing type 1 or type 2 diabetes for each individual HPAP donor. I am only going to focus on the genetic risk for developing type 1 diabetes. So here you can see within the boundaries, the red part of this density plot is nothing but the distribution of overall genetic risk score for developing type 1 diabetes for all of HPAP cohort. And the static lines are nothing but the location of an individual donor within this distribution. So you can see here clearly, and if the static line is on the left side of this distribution, the risk is low. And if it's on the right side, the risk is high. So again, I'm going to repeat, HPAP49 was the donor that risk for this donor is extremely low, less than 15% of the HPAP cohort. Now, if you scroll down, HPAP50, which was behaving more like a type 1 diabetic donor, has an extremely high risk, higher than 90% of the HPAP cohort, high risk of developing type 1 diabetes. So clearly, we saw something that the hormone secretion data was able to detect, some physiological difference. And and this genotypic data is able to reinstate and explain some of this difference, the reason behind some of this difference. But now I am interested in looking at potential manifestation of this physiological difference at a more molecular level with the help of gene expression data. So for that, I'm going to use the HPAP's single-cell RNA-seq data. So for that, I have opened the Cell by Gene portal, which is again another interactive portal also available on PancDB for our single cell RNA-seq data. Here, you can see an UMAP, which is showing you the distribution of all of the different endocrine cell types. For example, alpha cell. As you can see, as when I hover over these areas, they get highlighted. And then beta cell. I'm interested in beta cell, which is in the bottom right corner of this plot. So, and that's what I have gone ahead and selected. I've only selected beta cells, the single cell RNA-seq data for beta cells. As you scroll down, you will notice that I have only selected the single cell RNA-seq data for the two donors that I had previously selected, my donors of interest. HBAC49, that was more normal, and HBAC50, that was more like a type 1 diabetic donor. So if I hover over, you can see the distribution of cells for these two different donors. HBAC49 is showing clearly 2,800 cells, and HBAC50 has fewer cells, around 2,200. And another thing you can see that if you look at the color that's representing beta cells, this neon pink, HBAC49 clearly has a far more beta cells compared to HBAC50. Now let's look at the insulin gene expression for these two donors. The neon green areas are basically little to no insulin gene, are areas with little to no gene expression, and the non-green areas like here are higher insulin gene expression. So if you come over here, you can clearly see for HBAC49, which was more normal, has extremely high insulin gene secretion in 2,800 cells, whereas HBAC50, which was more like a type 1 diabetic donor, has extremely low insulin gene secretion. So again, let's recap. So we observed something phenotypically. We were able to explain that briefly in genotypically. And we dive deeper at the molecular level and look at insulin gene secretion and clearly see that there is something causing the difference in islet secretion in these two clinically healthy donors. So this brings me to the end of my live demonstration, but let's go ahead and recap everything that we presented today. So I was able to show you that as someone who is not a domain expert who does not deal with this data set, this islet physiology data, and someone who has never done single cell RNA-seq coding, I had a question and I was able to use the data and answer some extremely important questions without even downloading a single file. I also was able to show you how accessible all of the raw data is on PancDB. And I hope that this demonstration that Jeff and I gave would encourage you to engage with not just this data set, but PancDB as a whole. And all of the different interactive analysis portals that we showed you today and the ones that I have not showed you. There are plenty as you can, and you can go ahead and explore them. In terms of updates to the portal, as Jeff mentioned, by the end of the year, we hope to expand this data analysis portal to more assays like oxygen consumption. We will be adding more normalization methods. And of course, right now we have only 72 donors, but we already have the calculations ready for about 80 more donors, and we'll continue to add more donors to the portal. And I would like to end by thanking the organ donors and their families, without whom none of this would be possible. Thanks to HIRN and NIDDK for their support. Thank you, John, Leila, and Joyce for organizing this webinar. I hope this webinar was educational for everyone attending. I would like to thank our PIs, Dr. Kessner and Dr. Naji, for the opportunity to be part of PancDB and HPAP as a whole. Special mention for Dr. Anna Gloin, whose team generated the genetic risk course that I went over. I have to thank the PancDB team that I'm a part of. We are a small team of software developers, Tongbo and Yimin, and a bioinformatician, who's Noah, and we are led by Dr. Babak Faryabi and Golnaz Vahedi, and we are primarily located at Penn. The physiological phenotyping core that Jeff is a part of is the core that develops and generates all of the islet function data set that Jeff talked to you about today. And they are led by Drs. Doris Stoffers and Nikolai Doliba. And all of the experiments are performed by these amazing research associates, Wei Chin, Andrea, and Jeffrey. Jeffrey also helped me pick all of these use cases today that I showed you. So thank you, Jeff. And this brings me to the end of our webinar, and we are open to questions. Thank you so much.
That's great, Nilanjana and Jeffrey. Thank you so much for a wonderful presentation and very interesting demo of the system. Do we have, maybe you can stop sharing now and we can take a look if anybody has their hand raised. Is there any questions in the audience? So you mentioned oxygen consumption. What other assays are you planning to add?
I think we are going to begin with oxygen consumption. We also have data from Vanderbilt that we need to format and process that would work for the algorithm that is used in this portal. So those are the two options that we currently have. And then
I would just add, sorry, that our perfusion protocols are slightly different, us and Vanderbilt, but they're complementary. And you can see that in the sense we add amino acids and at Vanderbilt no amino acids. They work under a euglycemic regime that is very normal blood glucose. We have G3 and G16.7, which are both low and high glucose. And so together, there's a wealth of information that can be obtained for an individual donor. And I think really that's the strength of HPAP as a program, is that it can accumulate all of this information, perfusion data, immune cell profiling, single cell sequencing, that we can really dive deep into one particular donor, for example, HPAP 49, HPAP 50, and really try to understand what is going on with their islets, what's the islet function like, what is the gene expression like. So I'll just leave it there. Thank you.
That's great. I see Marcella nodding, but John had his hand up, and then let's go to Marcella after that. John?
Yeah, that's great. Jeff, this was the question I was going to ask. We are in a data-rich world, and that's terrific because we have a lot of great programs out there that are providing analysis, and you have Joyce and Marcella who are online here. So if the highs and lows are not there, but the euglycemic profile is relatively the same, is that directly comparable? Or not, or is that protocol a little bit different? And
Sure.
And just comment that I really like that use case 4950 and this tool that you've developed where it allows you to compare to the entire cohort. Was that and the question there, was that cohort comparison tool that takes everything, or can you customize what you include in the cohort? So in other words, can you say, no, I just want to look at the no diabetes, or I want to look at just the type 1? So just two questions.
Sure. Thank you. I'll go over the first question. This is about the perfusion protocols that we have. And again, like I mentioned, slightly different but complementary. For example, the use of amino acids in our protocol as the primary stimulus of alpha cells. So we can get a pretty good sense of alpha cell function through the amino acid step. But what it does is it also primes the beta cells for further insulin secretion when we add glucose. The protocol at Vanderbilt doesn't use amino acids, but again, as you mentioned, a euglycemic starting point. And so having both of those together are nice. It's difficult to compare directly. For example, they have a G16.7 step, we have a G16.7 step, but those may give slightly different values because we have different starting points. And you can imagine that it's not just the absolute level of insulin secretion that determines the absolute level of glucose that determines insulin response, but it's also where you came from. So for example, the response from 5 millimolar glucose to 16.7 is going to be different. In other words, the rate at which you give the glucose matters here. So we have this information from both a hypoglycemic condition and also a euglycemic condition. So we can assess then what's happening under a hypoglycemic condition versus what's happening from a euglycemic condition. I think having all this data together, there will be people far smarter than I who will be able to synthesize all of the data and come to more meaningful conclusions. But I think that's the first step, is just to allow everything onto the portal so that we can do these comparisons. And as far as your second question, I forget. Sorry.
The cohort
Sure.
cohort customization.
Right. So the customization is all done through the filtering on the left hand panel,
Okay.
and as Nilanjana had mentioned before, you can gate by age, for example, 21 to 30 in the example that we had previously. And there's also this tagging function, which Nilanjana had briefly mentioned. So let's say you were interested in particular subset of those donors which matched the filters, you can tag those donors, and then those would become a separate subgroup, which you can then explore further. And then there were also some features to be able to download the data and then to analyze the data one after the other for only those tagged donors. So I think this tool would be very important for people who want to do a very specific comparison of just a couple of donors who share very similar characteristics, for example, but may have very different insulin and glucagon secretion responses.
Yeah, thank you so much. That's great.
All right, this is Babak. So I just add a comment. So we, as Nilanjana mentioned, we do have a filter option that selects per source of the data that could one use to separate, let's say, Vanderbilt data from Penn, if they wish, and then compare their data sets.
Thank you, Babak. Marcella, did you want to comment on the question? Marcella?
Yes, sorry, I forgot to unmute myself. So first of all, Nilanjana and Jeffrey, kudos to you on wonderful presentations, and kudos to HPAP team on bringing this platform one step further to be able to visualize physiology data sets live. We have a lot of in common between Vanderbilt and Penn perifusion protocol. But there are also some really complementary things that we do. For example, what is very relevant to type 1 diabetes is stimulation of alpha cells with epinephrine, right? And it was wonderful how our protocols really benefit one from another, looking into cohort of GATA positive of islets from GATA positive individuals, right? Demonstrating that the alpha cell glucagon secretion is aberrant at single autoantibody stage. So I'm looking forward to actually connecting Vanderbilt data set through a wonderful portal that we built with Joyce and her team as part of IIDP for community to benefit from visualization tools and overall outputs that can be downloadable from the two platforms. As you know, we are also capturing imagery of islets that go into perifusion to be able to look at spatial organization of endocrine cells, especially 3 primary endocrine cell types such as alpha, beta, delta cells, and link that to their overall secretory outputs. One thing that I noticed, you have 1,000 islets in your chamber. That's a lot of islets. And burning question is, how do you pick 1,000 islets from individual with type 1 diabetes? Because those are very precious. And we know that those preparations, as a rule, are less pure.
Thank you for that question, Marcella. And I want to take the opportunity to thank you personally for your group's discovery of the alpha cell dysfunction in type 1 diabetes. Everyone has thought of type 1 diabetes as purely a beta cell disease, but really what you have shown is that type 1 diabetes is a disease of both the alpha and the beta cells. And then Nikolai Daliba over in our group extended that to the GATA positive donors, again, with your help. And this was not possible without HIRN, without HPAP program, to be able to accumulate all of this data. So first, I want to thank you for that. In terms of the question, 1,000 islets is a significant number, and we know this. And this is essentially a function of our specific perifusion system that we require 1,000 islets. However, in the case that we don't have a sufficient number, for example, we have less than 1,000 islets, it turns out that we can detect insulin and glucagon release from as few as 500 islets. So for example, in type 1 diabetic donor islets, which are not so plentiful, that we can actually detect insulin and glucagon at that level. We have a radioimmunoassay, but we also have ELISAs that we can run to be able to detect ultra low levels of insulin release and glucagon release. We are hoping to improve, of course, always these systems so that we can use fewer islets. For example, we've collaborated with the BioRep company to have their Peri5 system, which we recently acquired, and we're able to measure insulin and glucagon secretion from as few as 100 islets. But of course, as someone who designs and builds perifusion systems, of course, we have to make sure that the parameters are the same. The flow rate is the same. We want to make sure that there's laminar flow, not turbulent flow. And so all of these things need to be in consideration when we upgrade our systems. But we are always looking to improve our methods, but still maintain the same regime so that we can compare, for example, HPAP1 to, let's say, HPAP 300 in the future. So we look forward to that, and we look forward to speaking with you, collaborating with you as we have done. Thank you very much.
If I may follow up, I see Anna on the call, and looking at how you are integrating genetic risk scores with islet physiology, I think that's really one step forward, and this is really at the forefront of the collaboration with the Joyce and our team at Vanderbilt related to human islets that are being distributed through integrated islet distribution programs. So I look forward to bringing the platforms together and for a community to benefit from very rich data sets, as you nicely described during your wonderful presentation. So thank you.
Thank you, Marcella.
Thank you.
Thanks, Marcella. I think we're at the top of the hour, but thank you again for a wonderful presentation. And we look forward to that too at IIDP for possibly some future collaboration across our platform. So thank you, Jeffrey and Nilanjana. Marcella, thanks for your comments. Anna, thanks for your contributions, and hope to see all of you again at a future webinar. Thanks very much for joining today. Bye-bye.
Thank you.
Thank you so much.